Is my screen visible to you all? Yes?
Yes, ma'am, it's visible, ma'am.
Okay. So in today's class, we will look into another topic, that is studying cell and tissue properties. So far, we have looked into various aspects of cell behavior, like what all growth factors affect cell growth, what are signaling processes are there, right? Then we looked into detail the cellular fate processes. But now we are going to look into some techniques that people use to quantify it, like to actually observe cellular behavior in their like tissue samples or cell samples. Okay, so we can broadly classify the cell or tissue properties into two. One is at the cellular level, and another is at the tissue level. Okay, so when you are trying to understand what is happening to the cell, people usually observe what is the cellular morphology. Okay, how the morphology is changing with different condition. Okay, and also uh, how the conditions brings in change into the cellular fate processes like cell migration, proliferation, differentiation, apoptosis. So we already know in theory about each of these topics, but when you are going to experimentally understand whether the cell is migrating or not, or how it is differentiating, like in experimental condition, we are going to see how it is observed. At the tissue level, cell and ECM make a tissue. So when a cell and ECM together have formed a tissue, we also rely on its morphological characteristics. So we observe morphology, as well as we try to see whether or not the tissue is able to give some function. So characterize its functions. So we can do it by using some unique biomarkers, okay, that is specific to that particular tissue, or we can use some key features like what is the mechanical property of the tissue, okay, conduction. So various tissues show electrical conductivity, etc. So if they are showing these kind of functional characteristics, then we say the tissue is properly formed. So it is important for us to understand each of these techniques that I'm going to discuss now, because you are going to make an artificial tissue using tissue engineering techniques. Okay, and this tissue is going to mimic our body's in vivo tissue. So you want to, and the cell that you are going to use is derived from the body only. Okay, if you are using autologous cells, or it can be allogenic or from a cell line. So you are trying to see whether in presence of some ECM, artificial ECM, the seeded cells, when given proper conditions, culture conditions, is able to mature and give functions as exactly as it is seen in vivo. Okay, so at each level, starting from the cell to tissue level, we have to characterize at every level. Then only we can say at the end that the tissue is successfully formed and is ready for grafting. Okay, so when you see this tissue level and cell level properties, a very important technique, that is microscopy, using various kind of microscopes, is very helpful. Okay, so we can do microscopy at the in vitro level, where the cells are grown outside the body in culture flasks, etc. Or we can observe the cells inside the body or tissue function inside the body, that is in vivo. So, like for in vitro, we use various kind of microscopic techniques like phase contrast, bright field, dark field microscopy, then fluorescence microscopy, electron microscopy. Whereas for in vivo, we do MRI, CT scan, etc. Means various kind of in vivo techniques are also there. So for in vivo, there are some invasive techniques or there are some non-invasive techniques. Okay. So first, let's look into what are the microscopic techniques that will help you understand your cell or tissue properties in vitro, means at the lab scale. The first is the phase contrast microscopy. So here, uh, it is also called DIC, differential interference contrast. So that allows visualization of features that are actually transparent when you are doing bright field. So if any one of you get some time, like free time, you guys can Google these various type of microscopy techniques like phase contrast, DIC, bright field, because each type of microscopic technique gives you a different information. Okay, then there is dark field. So phase contrast is another one, phase contrast. And all these kind of microscopic techniques you can do using light microscope. Means you have to illuminate your sample and you're illuminating it using visible light. Now advanced microscopes that are coming in the market today, there you can do all the 3 4 types of microscopy in one machine. Okay, so like in phase contrast here, it helps you to see very means those features, those cell features that is completely transparent, that is not visible under a lot under a large amount of light. Okay, so it circumvents the limitation of bright field microscopy. Whereas in dark field, here what happens, the background, in the image there's a background and there the subject, that is your cells. So the background is completely dark. That helps you to better visualize more features like the boundary features of your cell, etc. So each type of uh, technique, the phase contrast, DIC, bright field, dark field, there are multiple Google like articles available, then YouTube videos available. So I recommend you guys to see a very good videos like videos by Nikon, Zeiss. So these are the companies who make the microscope. So they have their own demonstration videos available on the YouTube. You can see and understand the principle of each type of techniques yourself. But the benefit for tissue engineers, or why tissue engineers should know about various these types of microscopy technique, is you are it will help you to visualize the cells. So as you can see here, the cells morphology, how it looks like. So you can visualize cells at different magnification. Like this image is taken at 28 magnification. Then you can use microscopic techniques to uh, monitor your cell's health under some assays, like some experiment that you have set up. So this is one experiment, like time lapse experiment they are doing. Like at each time point, they are seeing how the cells are migrating from both the ends. So this is all taken from phase contrast microscope. So it is able to show you the clear margins, the white margins, okay, that is formed on the either side. Okay, so but this limitation, there is one limitation of each of these kind of microscope, is it helps you to see your cells on 2D plates. Okay, so if you have a cellular monolayer, so there you can individually see how the cells look like at different magnification. And for cell culture, we generally use an inverted microscope, where the objective, the placement of the objective lens is at the bottom. Okay, so here is objective and the light is coming from the top. That is called inverted microscope. Another kind of microscope is there, where the objective is object means another microscope that you might have seen in general biology labs. Okay, or even like in other places, okay, where the cells, they are not working with cells. So they generally have their objective lens at the top, and the light is coming from the bottom. But for cell culture, since there is a say flask like this, and cells usually grow at the bottom. So if the light is falling on the top and the lens is there at the bottom, it helps you to see it. Okay, um, helps you to see better. But the other way around, the other way of other configuration will also work for cell culture, but generally it is not preferred. This inverted configuration of microscope is the preferred geometry that is used for cell culture and tissue culture techniques, and as well as tissue engineers who are working in this area have microscopes of this type in their labs. So as I was saying, there is one limitation of these light microscope techniques, right? That it helps you to see cells as on a that is grown on a 2D substrate. Also, you cannot you cannot distinguish the structural features like the plasma membrane or the nucleus when you see bright field or phase contrast or DIC. So in order to distinguish each type of structures, like you want to see how the nucleus look like, or the cytoplasm look like, or the cytoskeletal elements look like, then what we do, we tag these structures with fluorescent dyes. Okay, to tag these structures with fluorescent dyes. So these dyes have the unique property to bind to specific type of proteins that are only available on these type of cellular organelles. Like fluorescent green is generally used to tag the cytoskeletal element. Then white blue is used to tag the nucleus. Then there is rhodamine, okay, that is usually used to tag other cytoskeletal elements. So these are different kind of fluorescent dyes that people use to stain their cells, okay, and visualize the location of the nucleus, the cytoplasm, etc. So in order to see these samples which are stained with fluorescent dyes, you need another type of microscope, that is a fluorescence microscope you need, where instead of means the white light. So in the previous type of microscopy technique, you are using the white light, which is having all the uh, uh, means you are using the visible range that is from 400 to 700 nanometer, correct? But here, what you are using, you are segmenting the white light into particular types of wavelength, like 405 nanometer, then 488 nanometer. So these are lights. So 405 represents to blue one, this represents to green one. So the light is segmented, and at this particular wavelength, you are exciting this fluorescent molecules, okay, means dyes, the molecule of the fluorescent dye, you're exciting it, and it emits some, uh, and you're capturing the emitted light that is coming, okay. So when this kind of principle is applied, so it is possible only through a fluorescence microscope, where light of a particular wavelength falls on your sample. That wavelength of light gets absorbed by
Ma'am,
the, yes, yeah,
ma'am, some people are waiting for your request acceptance, ma'am, in the meeting.
okay, okay. So as I was saying that here in here what happens in fluorescence microscopy, you are first staining your sample, cellular sample, with some fluorescent dyes, okay, and these dyes are specifically marking the cellular structures like nucleus, cytoplasm, etc. And when you are like irradiating your sample of a particular wavelength, so 405 nanometer light, you're very much particular of the wavelength that is falling on your sample. So at that wavelength, your fluorescent dye will get excited. It will, the electrons will move to a higher energy band level. Okay, because of which it will, when it will come to ground state, it will give some emission. So there are condensers, collectors in the fluorescence microscope that is present to collect this emitted wavelength, and then it is processed and given, and it shows you in form of an image. So when this type of principle applies, you use fluorescence microscope. Okay, so it is a better technique in comparison to the above techniques like phase contrast, DIC, dark field, bright field. Why? Because this technique allows you to mark, specifically tag your cellular structures. So you can use it for image enhancing your enhance, because the cells under visible light, they look very transparent. Okay, so when you're using dyes like this, you're color marking the structures, which makes the image much more good, understandable to the reviewer, whoever you're sharing this image. So people use this kind of fluorescence microscopy technique to check the cell viability. So there is one assay called live and dead assay, where there is a specific dye that tags only the dead cells, and there's a specific dye that tags only the live cells. So you can just like stain your sample with both of these dyes, and when observed on the fluorescence microscope, you will get signals of red and green that will help you understand how many dead or how many live cells are there in your sample. Similarly, you can also use this technique to understand the mitochondrial activity, the pH of the cytosol, and other cellular activity. But there is one drawback with this technique, is the light collected from the photon at many planes of the sample, they get merged. Okay, because of which the resolution of the image doesn't is not that much. Okay, there's someone else waiting. So what could be solution to this? How to increase your resolution of the image, or better say, like this fluorescence microscopy, it works well on 3D samples as well, like tissue samples. But all the signal that is coming from the different planes of a 3D sample, they get merged, because of which resolution is not that well. So for 3D samples, the best technique is to use the confocal microscopy. Okay, so here what happens in confocal microscopy, you are again like staining your sample with some fluorescent dyes. All right, but the thing is, the microscope system is able to cut your sample into various slices. Okay, and from each slice, and this slicing happens in the z axis, and from each slice the microscope gets the signal, the fluorescence signal that is coming, and ultimately all of the images obtained from all of these slices, they get merged and give you a better image. Okay, so here a computer records the fluorescence microscope of serial sections, that is the stacking, that is called z stacking. Along the z-axis, the sample, the 3D sample gets sliced. Okay, and they get combined into a image, that is a 3D representation. Okay, so now there are different types of confocal microscopy available. One type is there is only a single laser is used, the single photon is also called single photon, and there is then two photon when two lasers are used. Okay, and there is multi-photon, like more than two. The benefit of different photons are using more than one lasers is shown in this particular figure. So here what happens, if you use only one laser, okay, and this is an objective lens, this is an objective, this blue color is an objective lens that is basically converging the light rays. But here when you're using single photon or single laser, then the depth up to which your light can reach into your sample reduces. Okay, so it is a single photon is good if your sample is not very thick and you don't need very deeper slices of your sample. But you should go for two photon microscopy, confocal microscopy, because it can reach to a deeper sections of your sample along the z-axis. So the signals you will get will be like from the deeper planes of your sample, and the information that will help you gain will be much more accurate. Okay, so confocal microscopy, you can use it with samples which are fixed on the glass slides, or you can use it on directly live cells, like the cells you observe under confocal microscope and then keep it into incubator for further culture. Okay, so various modifications are available today that helps you that helps that can be utilized as per your need. So it uses the two photon uses two laser beams. Okay, and they are uh, like they excite the fluorophore, that is the fluorescent dye molecule, in the deeper zone of the sample, which emits at a longer wavelength. So basically exactly what I just said, the benefit of two photon confocal microscope, how it is better from a single photon confocal microscope. And note, in confocal microscopes as well, we are using the fluorescent dyes, okay, to uniquely mark the regions. Like if we want to see particular cytoskeletal element or the nucleus, etc. The same dyes that I mentioned, like the white blue dye, then the fluorescent green rhodamine. So these dyes can also be used for confocal microscope. Okay, but this is a better technique for visualizing 3D tissue samples. Tissue samples. Cells are basically 2D. When you layer the cells, it form a tissue. This is an example of how confocal microscope is used in tissue engineering. So figure IJKL, here you will see the cellular, means this is taken from a research article, okay, from a research article, where they are trying to show how the cell is infiltrating from one region to another region. Okay, so this blue color section is showing the cells which are whose nucleus is stained with DAPI. Means it is another form of white blue dye. So this is a dye basically that is used to stain the nucleus. So they are trying to show how the cells is migrating into the scaffold. So the red section there is the scaffold, the artificial matrix. And over the period of time, they have taken the image and see through confocal microscopy, they are able to take image from different planes along the z-axis and able to show that how the cells are present at each layer. Okay, this white line is basically showing the interface. So in this way, you can use this powerful technique to suit your needs. Okay, so for tissue engineering, it is a very common technique that people use, because ultimately you are going to make a 3D tissue sample, and in order to show how the cells are moving or how their properties are changing, how their morphology are changing, then one always uses the confocal microscope. Better to use the two photon one, so that you can use it with your thick tissue samples. Okay, this is another example where they have used confocal microscopy in order to see how the scaffold looks like in different planes. This is plane 1, plane 2, plane 3. And when all these images are merged, they look like this. And they use this merged image to simulate, to through simulation, make this kind of a 3D model. Okay, so this is called 3D reconstruction. Means from the image Like from 2D images, you are making 3D models, and the softwares are available that will help you do it. So basically, since confocal microscope is giving you information from 3D structures, this kind of analysis is possible. So any queries so far? Any general queries that might have come to your mind, or anything that you want me to repeat? No? Okay. So another very powerful microscopic technique is the electron microscope. So instead of light, okay, we are using electron beam here to illuminate our sample. The benefit of using this technique is you are going to get very good resolution, like up to zero 0.1 nanometer resolution. Okay, and there are various forms of electron microscopy. If the beam of electron is falling only on the surface of the sample, then that form of electron microscopy is called scanning electron microscopy, or SEM. If the electron microscope is going to pass through the sample, then it is going to be called transmission electron microscope, or TEM. And all these microscopic techniques, they happen or they are performed under vacuum conditions. Okay, air cannot interfere with the electron molecules that is coming from your source. Okay, so but there is another form of electron microscopy that can be done at atmospheric pressures, that is called environmental electron microscope. And this SEM and TEM, for these two techniques, you have to make sure that your sample doesn't contain any water. So we have to dehydrate it as well through some treatments. So when you are trying to see how the cells have aligned on your scaffold, okay, or you want to see how the cells have infiltrated into your scaffold sample, okay, or just you want to see the what is the porosity of your scaffold, because you need pores for the media or oxygen to diffuse to your cells. So you want to see what is the porosity. So these kind of questions are answered through SEM and TEM techniques. But when you do it, you have to make the samples dehydrated. The cells are basically made dead, and you cannot reduce those samples. But if you can do it under environmental conditions, then the process becomes easier. So this environmental SEM I have not seen, but this SEM and TEM techniques is very well available in all research institutes, and people are routinely using for various type of applications. Okay. So here's some images of how the like SEM and TEM are used to make images. So this is a face of Drosophila that is taken through SEM. That is, electron beam is only falling on the surface, so uh, so you are able to understand very finely the structural features, okay, of the face of Drosophila. You can even see the fine hairs. See how fine resolution you are able to get through electron microscopy. Similarly, this is a mitochondria, okay, that is present inside the cell. And through TEM, transmission electron microscope, where the electron beams is made to pass through a cell, you are able to see the organelle mitochondria and how it looks like. Whereas these two figures, figure this and this, here they are trying to show, this again taken from a research paper, where they are trying to show how the cells have aligned on different types of nanofibrous scaffold matrix. So here the fibers are aligned, like not exactly aligned like you have to keep it side by side, but they're aligned. Means you can see most of the fibers are like running in the same direction. But here the fibers are random, like forming a mesh. So they are trying to show this. So this is the cell. Okay, this is the cells. So they are trying to show how the cells have changed their morphology, like they extended their lamellopodias and grabbed onto the fibers. So this is another cell. This kind of like morphological changes that is happening on different type of scaffold geometry, you can see through SEM. So this is a SEM image. These two are the SEM images because the electron beam is falling on the surface. Okay. So now you have understood how you can use various kind of microscopy for understanding like cellular morphology, cellular structures at different resolution, at different depths for 3D or 2D samples. Okay, and these are all very, very much useful for in vitro studies. Okay, we have not gone yet in vivo studies. Now this is up to the cell, but when you're also studying the tissue, means you have made a tissue, artificial tissue, you need to understand how the cells are aligned in the tissue structures, what kind of ECM proteins are secreted by the cells. So there you need to analyze the tissue as a whole. So how you will do it? So this area has a separate name actually. This is called histology. So you must have heard of histopathology, where they try to understand the tissue characteristics of diseased tissues. Okay, but this is just a sub area or subclass. But histology is a very, very big area where they understand how the tissue morphology is changing with different conditions and using microscope. So the samples are basically stained using different kind of stains that is visible either through light microscope or fluorescence microscope. Okay, so you can use two types of stains. Either you can use classical stains, which are visible under light microscope, or you can use the technique immunohistochemistry, where either this works on either light microscope or fluorescent microscope, depending on what kind of molecule you are using in histochemistry, in immunohistochemistry or ISC. So we will look into detail of each kind of techniques. Okay, so let's look into some types of classical stains. So in the market, there are various kind of stains available that can be used to mark either ECM components or the cellular components. So these are some examples, like Alcian blue. It is used to it is used to mark the GAGs, glycosaminoglycans, that is abundant in cartilage. And the image something it looks like this, the blue color. Then there is alizarin red S. It is used to mark the calcium. So in bone, a lot of calcium is there in form of hydroxyapatite. Okay, so you can see this calcium deposition on the bone, and this red color spots is due to the alcerin redness. Then there is Congo red amyloid fibers. Okay, that is used to tag the amyloid fibers. Saffron O, again for GAGs. Then von Casa, it is for the phosphates. Okay, so hydroxyapatite also have calcium. This is actually a complex molecule that is composed of mainly the calcium group and the phosphate group. So you can check the bone mineralization either with alizarin red or von Casa. Then there is Sirius red that is used to tag the collagen that is abundantly present in bone, cartilage, muscle, etc. So these are some of the stains that one can use to visualize different ECM components in a tissue. Now you can also use some stains that are easily available for visualizing the cellular components, like DNA, okay, then heparin molecules, histones, glycogen. So these are all produced by the cells. Okay, so like you can use methylene blue to stain the DNA that is present inside the nucleus. Then toluidine blue that is used to stain the heparin molecules that is present in the mast cell granules. Then hemotoxylin, you must have heard about HNE staining. hemotoxylin and eosin. So hemotoxylin is used to stain the nucleus, and the nucleus has the histone proteins. So similarly, these kind of stains are available for staining different kind of cellular components. So major advantage is they are easy to use, easily available, okay, and the protocol for staining is not very complicated. With practice, one can easily do it, okay, and you don't need a sophisticated microscope to visualize your structure. You can use a simple light microscope under bright field, okay, and see the colors, how the and through color you will be able to understand the cellular component as well as the ECM component. But it has some disadvantages. So one disadvantage is that the binding interaction between the dye and the cellular or the ECM component, it is non-specific. So mainly charge-based binding takes place. So it might be possible that your dye can bind with some other structure and give you false positive results. Okay. Then when you are using these dyes with a scaffold, okay, and say you want to see how much calcium content is being secreted by the cells on your scaffold, and unfortunately your scaffold also has some calcium in it because of the polymer backbone or something like that. So the dye that you're using is going to bind with the calcium that is present secreted by the cell as well as that is present on your scaffold. So you cannot distinguish or you cannot quantify how much calcium is secreted by the cell. So this kind of limitations come into place, and then you have to play a bit smartly and think how to use these kind of stains for your purpose. This is another example that shows you how it can become a limitation when you're using these stains with different kind of scaffold. So for bone tissue engineering, the scaffold that are used, they are largely made up of hydroxyapatite based ceramics. Okay, and these ceramics, these hydroxyapatite, if you search in Google, so you will see calcium and phosphate groups are there. And these ceramics, they usually take up the alizarin red and one casa staining. So it when you use this kind of staining for this kind of scaffolds, it it makes the observer, the experimentalist, puzzled because he or she cannot is distinguish very clearly the cellular and the acellular constructs. Okay, so the example that I just gave before this is similar to what is written here. Okay, now you can use the stains, classical stains, to mark or to understand your tissue morphology, but you can also use another technique called immunohistochemistry, which is based on antibody and antigen interaction. So it is a very powerful technique that exploits the specific binding between the antibody and antigen to detect and localize specific antigens in cells and tissues, and they can be most commonly detected either with light microscope, or if you're using fluorescent dyes, then you may need to use the fluorescent microscope. So as the one disadvantage of classical stain was non-specific binding, so that is being solved if you are using immunohistochemistry. So before you analyze or before you wish to use this technique, you have to first fix your samples. Okay, and fixing is done usually uh, using formalin, that is basically 40% formaldehyde solution. Okay, the tissue or your artificial tissue constructs, they need to be fixed, and after that only you can do the subsequent steps. So what steps are there? First step is antigen retrieval. So what happens due to formalin fixation or the formalin based fixation of your tissue, the key antigens to which your antibody will bind, they get masked. So you have to do some treatments, enzymatic or chemical treatments or heat treatments, in order to expose those antigens. Then you will add the primary antibody. So you have to purchase the antibody specific to the antigen that you're looking for. So if my antigen is collagen, and I have to look for anti-collagen antibodies. Okay, then people use a secondary antibody or directly use primary antibodies. Secondary antibodies actually, if you're using secondary antibodies, then the signal that you are going to get from the dye or the fluorescent molecule is can be achieved at a maximum level even with small amount of antibodies, because these antibodies are very expensive and they come in a very small amount, like few micrograms or nanograms. So that is why like a primary antibody on top of which a secondary antibody is used, and the secondary antibody is specific to the primary antibody, means it can bind to the tip of the primary antibody. And to this secondary antibody, there is some conjugated enzyme or some conjugated fluorescent dye, okay, which in presence of some substrate, your enzyme will act on that substrate and release some colorogenic product. Or if there is fluorescent dye, in presence of particular wavelength, you will get some fluorescent emission. So this is the basic principle. Now, as I said, you can either directly use the primary antibodies, where the primary antibody is conjugated with some enzyme or fluorescent dye, but the main disadvantage is there is signal is very less. Okay, so the signal is less. Signal means the amount of color product or the amount of fluorescence that is coming out. If that is less, you won't be able to see it under the microscope very clearly. So there will always an ambiguity whether whatever you are seeing under microscope is actually the correct structure or not. So in order to increase your signal amplification and also get rid of this ambiguity, people use the indirect method where secondary antibody specific to the primary antibody is used. Now you can use various kind of labels, means the conjugates. You can use various kind of conjugates like enzymes. For example, horseradish peroxidase, HRP enzyme, or alkaline phosphatase. So these enzymes, they react with the reagent like, for example, diaminobenzidine, and give some colored product. Or you can use fluorescent molecules, okay, as a label, and but here you require a fluorescent microscope. So this is the image of what I have just explained in the previous two slides. So this is your, say, tissue or cell sample, okay, and you want to specifically visualize whether a particular type of protein is synthesized by the cell or not, or is it present in the tissue or not. So here in the image 9, people are trying to understand whether the presence of collagen type 1 is there or not. So they used the immunohistochemistry technique where the secondary antibody is used, that is indirect immunohistochemistry is used. The secondary antibody is conjugated with HRPO, horseradish peroxide, right? Horseradish peroxide. So HRPO enzyme, and this enzyme in presence of the substrate DAB, it gives a brown colored product. So that is why you see the collagen is color stained in brown. Whereas in immunofluorescence, means we are using a fluorescent dye instead of an enzyme to tag your antibody. So in immunofluorescence, here, although this is a direct example, but indirect can also be done. In direct, you are only using the primary antibody conjugated with the fluorescent dye or enzyme. In indirect, a secondary antibody is used. So here the fluorescent tag is there, you give some light of a particular wavelength and you get a fluorescent signal. So here in this image, collagen type 4 is stained with anti-collagen 4 antibodies, means antibodies specific to collagen type 4, and that helps you understand the distribution of collagen type 4 on your tissue sample. So here it is done on mouse embryonic mammary gland. Okay. So these are the few techniques that can be used to understand your tissue or cell morphology. And all these techniques were is possible in vitro. Means you can analyze your samples in vitro conditions, that is outside the body. But if you want to understand how the cells or tissues are present or organized or how they're functioning, then you have to opt for the in vivo techniques. So simple technique to see what is going on inside the body is through imaging. Okay, so there are two types of imaging methods. One is the non-invasive one, another is the invasive one. Okay, so non-invasive method use the ultrasonic waves, that is USG, that like often people do of their abdomen in order to see how the organ function. Then there is X-rays. Then you can use fluorescence microscopy as well, that is non-invasive. Then CT scans. Okay, then MRIs, then nuclear medicine scans. So these kind of non-invasive techniques are there. So here, like in CAT scan or MRI, lights of various wavelengths and electric fields and magnetic fields are applied to get image, and these image can be used to make 3D models, that is 3D reconstructions can be done. Whereas in invasive method, here you are using probes, miniaturized probes, where cameras are present and it is inserted into your body through some incision. Okay, and then people observe like what is going on. So you must have heard of endoscopy, right? So laparoscopy. So these kind of techniques are like invasive techniques, invasive imaging techniques, where the patient or the animal has to be given anesthesia to so that he or she doesn't feel any pain, and inserted the camera probe. Okay, so this is again an image taken from paper, where this is an image of a micro CT, that is a CT scan but done at a micron level, means resolution is of micron level. So this is basically a mice, and through CT scan we can see how the bones, the vasculature or the organs are present inside the body. And this is the CT scan of different organs, like this is for kidney, this is for brain, I think. Okay, this is for brain, and this is like showing how the vasculature is present. Okay, so you can like this use these powerful techniques in vitro as well as in vivo in order to understand how the cell or the tissue is properties are changing, like the morphology, function, etc. So any queries so far?
Ma'am, one doubt, ma'am.
No doubt.
Ma'am, while talking about immunohistopathology, you said for a
Okay.
particular antigen, use that antibody, right?
Yes,
Ma'am,
right.
but for diseases, every time a microbe will get mutated, right? Uh, while that time it will produce a different antigen, right? Uh, but why we need to buy a same antibody and we trying to fuse with the antigen, ma'am? How it will be bind?
Uh, okay. So whatever you have studied from immunology, I think you're confusing here. So here the antigen is not produced by the body in response to some foreign. Okay, means antigen is not the microbe that is coming into your body. It is means anything protein based, anything. So antigen is collagen, or antigen is any cell secreted protein. And if you remember, antigen has epitopes and paratopes, right? So specific to that epitope, people make monoclonal antibodies through techniques like hybridoma technology. So we are using that. We are like In hybridoma technology, we are giving, say, collagen type 1 or type 2, introducing inside the body of a mice, and that the mice perceive it as foreign, and in response to it, antibodies are produced. Okay, so we purchase the antibodies that is produced by that particular mice, and it is applicable to us because we want to see collagen type 4 or type 1 in our sample. Get it? Means any protein produced by the cell or present in the tissue is acting as an antigen, and specific to that, uh, like antibodies are available. Understood?
So here antigens means proteins that are normally present in the body.
Yeah,
It's not like
exactly, exactly,
others. Okay.
exactly. Okay.
Okay, madam.
Any other queries? All right. So now we will move to a different area actually. Okay, in different area. So now we will understand how to uh, track the cellular fate processes, like how to say whether the cell is viable or not, means cell is living or dead. Then how to quantify the cell differentiation or cell proliferation, cell migration. Okay, so cell number. So we will now look into these aspects, the quantification of the cellular fate processes, and observing it or quantifying it at the experimental conditions. So first, very general thing that you will have to do when you're trying to make a tissue construct is define how many number of cells you want to put on your artificial matrix. Means you need an exact cell number. So how will you count the cells? That is what I'm going to discuss. So two techniques are possible. You can do it through direct method, which is non-destructive in nature, or you can do it through indirect method, where you have to lyse the cells, that is destructive in nature. So in general, people use the first one, the direct method, and for that people use these two types of machine. One is called a hemocytometer, another is called a Coulter counter. So in hemocytometer, the accuracy of the number of cells, it depends on the person who is counting, because you're manually going to count the cells under a microscope. So you use a hemocytometer device that looks like this. So this is a device. Okay, this is a device, and on hemocytometer, 5 by 5 matrix is there, and so so this matrix can be divided into 4 quadrants: 1, 2, 3, and 4. So these are 4 quadrants. So what we do on the, this is the device, hemocytometer device, and on the device, this quadrants are etched. Okay, under the microscope you can visualize where the lines are, and you pipe it out very few microliters, like 10 microliter of the cell suspension solution, and put a cover slip on it. So your cells are now fixed, like temporarily fixed. They cannot move. And because a vacuum is there, the vacuum is created, the cells cannot move. The cells under microscope, you will look, the cells will look something like this. Okay,
Ma'am, you are not
and
presenting.
my screen is not visible.
Yes,
Can
ma'am.
others confirm?
No, ma'am,
Ma'am,
it's
yes,
not
this screen is
visible.
not visible.
Okay, so let me reshare my screen
Ma'am,
again.
also Nidhi Priya is waiting in the lobby to get in this meeting.
Okay, okay. Am I like, is my screen visible now? Is my screen visible?
Yes, no, ma'am.
Is it visible now?
Yes, ma'am.
It's visible, right? So what was the last slide you guys saw on the screen?
I'm 2 slide previous. Black one.
So this is the last slide you saw. Or this one you saw, I think.
Yes, ma'am.
This one you saw, no? So from this slide onwards, my screen sharing stopped, correct? Okay, fine. So I was going to, like I said that we are not going to understand how to understand whether or not the cellular fate processes are undergoing, means whether cells proliferating or whether it is dividing. So you have to know that under experimental conditions. You have to perform some experiments or use some techniques or methods to say with confidence that the cell is differentiating or cell is proliferating or cell is live, it is viable. So very first thing is you need to quantify or you to quantify how many cells you need for your tissue engineering application. So you make a scaffold out of, say, a material, a biomaterial, and on that scaffold you have to add some cells. So how many cells? Okay, so what is the cell density? So in papers you must have seen people define some cell densities, some number of cells, xyz number cells per ml. Okay, so the unit people use is cells per ml. So how did they come to that number? So people do it via two techniques, either the non-destructive direct method where devices like hemocytometer or Coulter counter is used, or people use a destructive method where the cell is lysed completely. Okay, so in hemocytometer I was going to explain the structure, how the hemocytometer's device looks like. So this is a hemocytometer device. This is a hemocytometer device, and on this device you pipe it around 10 microliter of cell suspension, uh, and you put a cover slip on it. So after putting the cover slip, the cells become stationary under vacuum, and when you see it under microscope, you will see a 1 by 1 square matrix where there is 5 square by 5 square. Okay, now in general it is divided into 4 quadrants: 1, 2, 3, 4. So we count cells in each of these 4 quadrants 1 by 1. Now if you notice, in each square you will see there are more squares, like again 1, 2, 3, 4, 4 by 4 squares. So these squares helps you to count, to understand how many cells are there inside the boundary. We don't count the cells that is outside the square boundary. So we count manually by observing under microscope the number of cells that is present in each quadrant, 1, 2, 3, 4, and then we take an average of it, and then we use unitary method to calculate the number of cells per ml. Okay, so this is a straightforward calculation that is like routinely used in almost all cell culture labs to count the cells. Now those right, sorry. Now some labs Now, some labs also use automatic cell counting machines. Okay, so one such example is a Coulter counter. So there, what happens? The machine does the work for you. It counts the cell. You supply some microliter of cell suspension. Like here, this is the loading area where you load the some device where you add some drops of cell suspension and insert it into the device, and the device can count the cells for you and display the number. But this kind of machines, the cost is more. Okay, whereas the use of hemocytometer is the cheapest one, cheapest technique. So here, what principle plays here means in culture counter. Here you use electron signals, okay, and where each of the cell is treated as a particle, and they basically detector is there that is able to help going to detect how many cells are there. So through electrical signal, each cell is charged and fallen on a detector, and this detector is going to count the cells and show you the final cell number. But there is some limitation with this technique. One technique is one limitation is the cell suspension that you're making. There should not be any cellular clumps. Okay, so if there are more number of clumps, the cell suspension is not made uniformly, then the number will be wrong. But in hemocytometer, if you are very much practiced person and you have done it hundreds of times, then most probably you are going to get like accurate number of cell numbers every time. Okay. So hemocytometer comes with practice. Those who are a bit lazy, they can go for Coulter counter, but again, there can be some error. Okay. Now there are various indirect or destructive methods to determine the cell number. One, there are methods that measure the DNA content, the protein content, metabolic activity of the cell using some fluorescent dyes or colorimetric protein binding dyes or enzymes. Okay, in order to understand, means indirectly understand how many cell number is, how many cells are there. So DNA, if it increases, the cell number is going to increase, right? So this kind of logic, indirect ways are there to measure or determine the cell number. But these measurements are prone to error because there are cell to cell variations. Like in some cancer cells, uh, these cancer cells shows DNA to cytoplasm ratio that is high. Okay. Also, the hepatocytes, they are often multinucleated. From one cell, you will find more than one nucleus. So the DNA content that you are getting, you cannot accurately determine how many cells are there. You will get more number of cells, but in actual there was not that much cells. So this kind of errors can also happen using indirect or destructive methods. So now you have determined how many cells are there, okay, and you know that you are going to seed say 1 lakh or 1 million cells. So for determining 1 million cells, you have counted your cell suspension. The next is you have seeded your cells on your scaffold. You have to ensure whether the cells are viable or not. Is your growth conditions good enough to keep the cells alive on your scaffold? So you then have to go for cell viability assays that helps you understand whether the cells are alive or dead. So again, there are various kind of methods available that uses the cell morphology, integrity of cell plasma membrane, then metabolic activity, enzyme specific activity, presence of DNA, etc., as important markers to assess the cell viability, means that the cell is alive or not. So um, we use generally the metabolic activity as a important criteria to show the cells are viable. Okay, and we can also use trypan blue stain that can stain the dead cells. So you can actually visualize again the dead cells and the live cells. So sorry. So is my screen visible? So in this image, this is a cell suspension mixed with trypan blue, and it is pipetted over a hemocytometer. And under microscope, the cells which are deep blue in color, they are basically dead cells, where the other bright red color cells, they are live cells. So you can actually count in this way how many dead or how many live cells are there using the stain trypan blue. And there are similarly other kind of stains or assays are available that helps you understand whether the cells are living or dead. So any queries so far? Okay, can any one of you confirm whether or not my screen is visible?
Yes, ma'am, it is visible.
Okay, okay, fine. So next uh, thing is, if your cell is of adherent type, means it needs a substrate to grow on, then you can also visualize whether or not cell adhesion has happened properly or not. You can visualize through microscopic technique, or you can quantify it, okay, by measuring the amount of shear stress required to detach the cell from the surface. So this is an image, microscopic image. So this is basically a fluorescent microscope image where the fluorescent dyes like DAPI or Fitzpalloidin or like rhodamine, these kind of fluorescent dyes are used to specifically target the different cellular structures like the nucleus, the actin, F-actin cytoskeletal elements, then the vinculin. Okay, so if these structures, through these structures, and also you can see the cell morphology, how the pseudo lamellopodia they have spread. So when cell is adhering, this kind of spreading of the cell takes place. So from this type of microscopic images, you can confirm that the cell has adhered on your substrate properly. Okay. Now you might be wondering that why we are going to now see the cell proliferation. Why to quantify the cell proliferation, or the rate of proliferation of the cell? Because we already have the cell viability assays, right? So if the cell is living, it will definitely proliferate. So it becomes necessary when you're trying to study the stem cells. So what happens when the stem cells divide? The daughter cells, they can either like stop division, stop the cell division, like they can retain their stem cell characteristics and contribute to replenish the stem cell pool, or some of the cells they will continue cell division and give rise to differentiated cells. So when this kind of questions you want to answer, whether cell division has stopped, or up to what cycle, or up to what age the cell is still dividing, the number of times you want to see, then uh, the assays that are used to quantify the cell proliferation is very much helpful. So cell proliferation is correlated with the rate of DNA synthesis. Okay, it makes sense, right? So cell cycle happens, so there is an S phase where the DNA content gets doubled. So if we use some markers like labeled nucleotides, like fluorescent labeled nucleotides or radio labeled nucleotides, and added into the cell media, so that the cells take it up and incorporate it into their DNA. So we can then, as the cell is proliferating, we can then check under fluorescence microscope, or by giving radioactive waves, and see how the DNA is getting passed on from one generation to the next generation. Okay, and see up to which level the characteristic, stem cell characteristic, etc., is retained, or it is going to a differentiated state. You can even do lineage tracking. Very good example is lineage tracking. Means we want to confirm that from hematopoietic stem cells only the RBCs are formed. So you can actually tag the DNA using these labels and see whether it is passed on to the final mature cells or not. So this kind of assays, or this kind of questions, can be answered through this cell proliferation techniques that is used to quantify cell proliferation. Then, if you recall, I have already discussed about cell differentiation. Remember? So there we saw that using the technique of DNA microarrays or RT-PCR. So these techniques are very much useful in order to understand what all mRNA is being expressed or what all genes are available, okay, and they are expressed, means the down regulation, the up regulation of these genes, you can see it, okay, using the techniques like DNA microarray and RT-PCR. So in your free time, you can definitely watch YouTube videos in order to understand in more detail what is DNA microarray or what is RT-PCR. PCR you know, but RT-PCR, real-time PCR. So you can actually see videos, like ample videos are available. Then last is, we need to understand how apoptosis is taking place. This is another very important thing, because when people are studying cancer cells and they want to understand whether the drug is effective or not, and what mechanism, through which mechanism the cancer cells are getting killed, whether it is apoptosis or what kind of mechanism. So this kind of characteristics, a quantification of these characteristics are very much helpful. So uh, apoptosis is characterized by formation of microbodies. So if you see microbodies formation in the cell, which is which is a morphological study, means the cellular morphology is changing when apoptosis is triggered, then you can confirm, yes, your drug is effective, your cancer cells are dying through apoptosis. Also, there is the hallmark event of DNA fragmentation. Okay, so you can also fluorescently label the nucleotides by this reaction, nucleotidal transferase reaction, and see the fragmented DNA. You can do electrophoresis, etc. Okay, or you can see under fluorescent microscope the DNA fragments, or you can use this information that phosphatidylserine, which is present in the lipid bilayer plasma membrane, it flips out on the outer side from the inner side. So when it flips out, a molecule called annexin 5, it binds to this phosphosidyl residues. So this event only happens when the cell undergoes apoptosis, correct? So whether an annexin 5 is binding or not, you can check it, okay? You can tag this annexin 5 uh, using primary antibodies, and these primary antibodies can be conjugated with some fluorescent dyes, and you can selectively see which of these cells are apoptotic. So this kind of information can help you understand whether your cell is undergoing apoptosis or not under some defined conditions. Now, measurement of cell migration. So when we were studying the mechanism of cell migration, there we didn't cover how to actually see whether cell is migrating or not, or how to basically measure the migration of the cell. We only saw graphs, right, where people use lines to track the path of the cell, but how they actually do it. So when there are various assays available, so I will briefly explain each of these. First is the Boyden filtering transmigration assay. So here, what happens? There is two chamber. One is the upper chamber, one is the lower chamber. In the upper chamber, you add some cells, okay, and in the lower chamber, you add the chemoattractant. Means cell migration always happens in presence of some chemokine, right? Because cell is trying to migrate towards the source of this chemokine. You know already. So that is why a chemoattractant is present in the lower chamber. And after some time, they see whether the cells have transferred or transmigrated from upper chamber to lower chamber. But there is one limitation of this technique. This limitation is you cannot actually observe the cells during migration. You are able to observe the cells before migration and after migration. So during the migration, observation is not possible through this Boyden assay. So this limitation is solved by a scientist who made a chamber called sigmoid chamber. So here the geometry is different. Here we are not using two chambers. but here slide is used, plexiglass slide is used with two rectangular wells. So these are the wells, well 1, well 2. Okay, and they are separated by a bridge which is 1 mm in diameter. Each of these, one well is filled with media, another well is filled with chemoattractant, and cells. So this is the side. This is the slide. The cells are attached to a cover slip, which is then inverted and fastened to the slide. So this is the cover slip, and it has some say protein coating like gelatin coating, etc., on which the cells can adhere. So cells, once the cells have adhered on the cover slip, they, using the clips, they invert it. Okay, and they try to then see under microscope how the cells, like cells are on present on the top, like when you have inverted this clip, the cell is present on the top here, the clip will come here, the cells will be present here. So the media, in presence of media or in presence of chemoattractant, in which direction the cell is moving, you can actually observe it. When, since it's a glass slide, right, it is transparent, so you can observe it under a microscope. But again, the limitation is there, is the wells that is made is having rectangles, like rectangular geometry is there, so edge effect will come into place. So to solve it, another chamber is made, that is done chemotaxis chamber. Now here the same thing is happening, but only the wells are present in a concentric manner. So in one well there will be media, in another well there will be chemoattractant, and there is a bridge that is separating between the outer well and the inner well. And on the inverted side of the cover slip, there is cells adhered, and you can see under microscope how the cell is like migrating either towards the chemoattractant or towards the media. Next, another technique is there, that is called agarose assay. So here what is happening is agarose is poured into a chamber with a glass bottom, creating two wells. Okay, one for the cells, another for the chemoattractant. So some chamber is there which is having two wells, and onto which agarose is poured, and in one chamber or in one well the cell is there, and another this chemoattractant is there. Now the chemoattractant is basically a solution type of, so it can agarose is a hydrogen, so it can uh, like absorb water. So this chemoattractant solution, it can diffuse through the gel and create a gradient basically. Okay, uh, and in presence of this gradient, the cells that is present in another well, they will migrate through the agarose and will come towards the chemoattractant. So I have not seen this kind of a geometry, but what I assume is there will be some well like this, okay, some well like this, and in between there will be agarose. And so in one well there will be chemoattractant, and another well there will be cells, and this chemoattracting solution will diffuse through the agarose gel, so, and create a concentration gradient, here high, here will be low, and the cells in will sense this concentration gradient and will try to move towards the high concentration of the chemoattractant. So through the agarose you can actually see, you can use here confocal microscope in order to uh, how in order to see how the cells are placed in different z planes. Okay, because this is a hydrogen, this is a 3D structure. So this kind of assays are available. Other than that, you can also use other methods like oriented collagen gel assay, inclined plane migration assay, conjoint fibrin gel assay. So various kind of assays are there that will help you to quantify the cell migration process. Now, So any queries so far? All right. So we are almost towards the end of our discussion today. Now, in order to see how the cells behave in response to some mechanical force, you apply some force on it, and then you study what kind of mechanotransduction signaling takes place in the cell. So there are various techniques to apply some force on one single cell. So you can use AFM tip. AFM stands for atomic force microscope, and it has a very fine tip, and you can use a tip to create an indentation over the cell surface, or you can use a pipette to suck the cells. So this is like you are, so this looks like something like this. You can use tweezers, optical tweezers, to compress the cells, or you can use shear stress like flow conditions to detach the cells. So under these kind of methods to exert some external force on the cell, you can quantify how the cell is responding to different mechanical forces. There can be some other, so these all techniques that I said, these are all passive techniques. Means you're not exactly, what do you say, measuring the mechanical properties directly. Indirectly, you are going to do, means like for example, detachment by shear stress. If this technique, if we see some flow rate, through some flow rate, you are going to flow some fluid over the cell, and you're using the flow rate as a measure uh, to say that at a very high flow rate, the cell is, some percentage of cell is detaching. You're not exactly using the term shear stress. Due to flow of the fluid, some shear stress is going to be applied. So this is a passive way of quantifying the effect of the mechanical forces, and thus the measure the mechanical property of a cell. But there is an active way as well. Means you culture a cell on a substrate, and this substrate is attached to some sensors. Okay, like load cell, etc. Some sensors are there which is capable of like translating the cell's adhesion forces into some electrical signal, and then you can see it on the computer. So this is an active way. Directly, you are getting the information from the cell, but in the passive way, indirectly, like through AFM tip or through pipette tips or through tweezers, you are applying some forces, and depending on how much force you are compressing, okay, or depending on how much flow rate of the fluid is flowing, you are going to say like this much strength, which this much strength the cells are, your cells have added on the surface. Then for tissue engineers, it is much more important to understand the mechanical properties of their tissue rather than this individual cells. Okay, because in our body there are tissues of different strengths. Like the bone is the hardest tissue. Then there is soft tissues, uh, and there are tissues that are like the brain. Brain is very soft, but and there are cancer models. So when tumors, you see the mechanical property of the tumor, means the Young's modulus is very high in comparison to this tumors formed by the clump of cell, in comparison to a clump of cell or a ball of cell or a spheroid of cells. So through the difference in mechanical properties, you can distinguish the tissue type. So it is then important for us to measure the mechanical property of the tissues. So there are again two techniques. One technique is through passive way, another is through active way. So active way, you directly use some machine. It is called mechanical testing machine that generates some that through which you apply some force and see and apply some force to generate a strain on your material, and response to which you calculate the stress. So this kind of active techniques are generally used, but there are passive techniques as well, like the viscoelastic properties, means like the pulsatile flow actually. So the blood vessels, they are always pulsating, right? Because our heart is like pressure is increasing and decreasing, right? Systolic and diastolic pressure. So because of which there is a pulsating motion in the blood vessels, and that is that through that you can quantify at with how much stress the blood vessel is able to contract and relax. This is measured to understand the mechanical property, the Young's modulus of the blood vessel. But generally, for tissue engineering, we use the active method where a mechanical testing device like this is used. So you can apply either compressive force, like in this example, they have used a compressive force, or you can use a tensile force, where from both the ends you are pulling the sample from both the ends. Compressive force, from both the ends you are compressing it. Okay, and there are different type of tests that is possible through this kind of mechanical testing devices. So that's it for today's discussion. Okay, so we have actually covered a lot. We have actually completed one entire section of our course, that is understanding the cellular and tissue properties and how you do it through various characterization techniques. So if any one of you have any queries related to whatever we have discussed today, please let me know.
Ma'am, in EMF, ma'am. Ma'am, in EMF, there will be cantilever, right, at the tip.
Yeah, correct.
We call the tip as a cantilever, right?
You're correct. You use the correct term.
Yeah, and it will be moved on the cantilever will be touching the cell, and it will be moved from one region to another region, right? This is how it will be happen, right?
Correct. It will create,
Okay.
as it is moving, it is going to as it touches the cell, it will create an indent. Okay, you can control with what amount, means how deep the indentation will be, means you can control the pressure that the cantilever can apply on the cell surface. Okay, and you can see whether the cell membrane is getting ruptured or not at this pressure. So you can then say ki this much is the strength up to which the cell can sustain that pressure. So this kind of analysis can be done.
And the topography.
Topography,
And also,
don't use the word topography, means you can simple term is surface, cell surface. Okay.
surface. Okay, cell surface. Okay, ma'am.
Anything else? Any other queries? All right. So if there is no queries, I think my screen sharing has stopped. Correct?
Yes, ma'am.
Yeah. So any one of you have any queries, you guys can let me know later as well. And did you guys see your quiz 6 marks? And have you guys seen the solution? Is there any queries related to that?
No, ma'am.
Okay. So let me then quickly take attendance and wrap up today's class. So Moldy is present. Vaishnavi?
Yes, ma'am.
Okay. Shambhavi?
Yes, ma'am.
Keetan? Okay. Janvi?
Present, ma'am.
Vani?
Yes, ma'am.
Harshita?
Yes, ma'am.
Nidhi?
Yes, ma'am.
So, all right. Um, Okay, I remember in previous 1 or 2 classes back, I think Molly asked me to share something about me, right? Can you, like, if you recall the things that you want me to share, can you repeat them? Because I have some time
Um,
today, I can maybe discuss. Yeah, what you wanted to know about me, I can share. No problem at all.
After you did your BTech from Viability, how you go to PhD without by clearing gate?
So, yeah, through gate marks, you have
Excuse
to
me, ma'am.
apply. Yeah.
Excuse me,
Yes.
ma'am. To mark my attendance as well.
Sorry, I'm not able to understand what you just said. Can you repeat?
Ma'am, attendance.
Excuse
They have
me,
not marked
ma'am.
my attendance.
Acha, acha, Aditya. Okay.
Yes,
Okay.
yes, ma'am.
Yes, I have marked your attendance, Aditya.
Okay, ma'am.
Okay. Harshita, you were saying something?
Ma'am, I was just repeating what you were saying. You weren't able to listen that.
Uh, uh, I have marked Aditya's attendance.
Got it, ma'am.
Okay.
Okay.
So after I did my BTech from IIT Durgapur, I directly applied. Means you can in IITs they allow you to apply for direct PhD, given that you have interest in research. Means you have to give some interview, and they call you for interview. And in some departments there is written exam as well. And when you clear both of these rounds, you can apply for PhD. But for PMRF fellowship, you have to apply. You can only apply once you get admission into one of the PMRF granting institutes. Like IIT Kanpur is one of those institutes. You have to first get admission there. Okay. And then there are modes, and there are two modes: lateral entry, direct entry, etc. So all this information is available on the PMRF website actually. And since I have to do some teaching work, that is why I contacted the people, means who are the, like, what you say, chairperson, etc. of the ISSS. So they allow PMRF students to conduct courses and complete their teaching requirements. And since my research area is in tissue engineering, my area, to be very specific, is I'm trying to make artificial skin construct using the technique of 3D bioprinting. Okay, so this is, in very short, this is my work, this is my thesis work. So since I have like extensively studied this area so far and still learning actually, all of these techniques that I discuss in our classes, many of these techniques I have done it, or I'm going to do, or like I have seen others do it. So I thought this is a very good platform, not only to share the knowledge on what I have gained, but as well as to revise, to learn a bit more. So that is why I floated this course, and I hope so far you have enjoyed. Okay, and if there is any queries related to anything that has been discussed so far, you guys can ask me. Even if I don't know the query, I'll give my best to find the answers to it. And maybe in next semester as well, I'll float some course related to some other topic. I don't know, but it will be available on the ISS website. So if any one of you is interested, you can also apply to that course as well. Anything
Ma'am,
else you guys want to know? Yeah.
yes, ma'am. Regarding PMRF, one doubt, ma'am. Whether
Hmm.
based on a thesis they will grant you, or already they will have some thesis, you need to take that and get a PMRF scholarship.
So there is some eligibility criteria. For first, you have to fulfill like the CPI, then the course credit requirements, etc. And then you can then screening happens. So at the department level, then at the national level, and in the national level is the main screening that takes place. Like if say our department sent around at the national level, and during application process you have to submit your thesis proposal. Means what area you want to work on or what research problem you want to work on. So this is for the students who are going through lateral entry, whereas students who are going through direct entry, they have to submit a SOP, statement of purpose, where they have to explain why did they join for PhD or and also what research areas he or she is interested to work in. Okay, why he or she is a good candidate for PMRF. So these kind of things they need to write. So depending on what mode, lateral entry or direct entry, your application process becomes slightly different. Okay. So thesis topic during PhD doesn't get defined immediately once you join. It will take some time. Okay, around 1 to 2 years, you will have to just study. You have to take coursework. You have to give exams. You have to go through training periods, learn softwares or learn experiments, learn techniques. Like I have learned cell culture only. Like it took me 3 months in my lab. Like the seniors train us. So a lot of time takes goes by just in training and learning. Okay, after that are you can actually you gain confidence to work on your thesis problem. So it's a long journey. PhD is a long journey. It's a life. It's not just a degree composed of few courses, but it's a challenge in itself. All right.
Okay, ma'am.
All right. So are you thinking for applying for PhD in future?
No, ma'am. No, ma'am.
No. All right.
I'm
Good.
preferring for GATE. That's why I asked.
Okay, okay. So through GATE you can also get jobs like in PSU etc. But if you are from like you guys are from biotech, I know. But when I was in your stage, like just completed my BTech, I also had an option to go for job. I had a good GATE score as well. But the thing is, no PSU no core PSU takes biotech GATE. Since we are non-core, right? So it becomes difficult to get into PSU through biotech GATE score.
No, ma'am. I'm aiming for MTech, ma'am. MTech biomedical engineering.
All right. So yeah, that also. But if for IITs, you need to get a very good GATE score.
Okay, ma'am.
Biomedical engineering is a very like the tissue engineering comes under this area. It's a very interesting topic and has direct application to changing people's lives. So yeah, go for it. If you get opportunity, go for it. It's very interesting. Okay.
Yeah, okay, ma'am.
All right. Let's wrap up today's class. I'll see you guys next week. So next week, most probably there will be quiz and other things. I'll let you know either through message or WhatsApp. All right. Bye bye. Good night.
Bye, ma'am.
Thank you, ma'am.
All right.